Hello and welcome, everyone. It is December 3rd, 2020. This will be a Davis Complexity Group webinar with Andy Lomas entitled Morphogenetic Creations. We're really looking forward to it, and it's going to start in 25 minutes. So until then, here is some awesome complexity music by Michael Garfield. And there's details on the sides of this image if you want to learn more about Michael's music. So Michael, let's hear your music video, and the rest of you will look forward to seeing you in a couple of minutes for Andy's talk. Hello, everyone. Thanks for coming out to the complexity event that we are having at Davis Complexity Group. This is Daniel Friedman, and I am very pleased to be introducing our speaker, Andy Lomas. Andy Lomas is a computational artist, mathematician, and Emmy Award-winning supervisor of computer-generated effects. His artwork explores how complex structural forms can be created emergently by simulating growth processes. So certainly at the intersection of art and science. Currently, he is based in London, so it's awesome to have an international complexity group speaker where he is developing his art practice as well as a lecturer in creative computing at Goldsmiths, University of London. So thanks again, Andy, for coming to speak for our group. We're really looking forward to your talk, and please just take it away. Hi, I actually do not hear you yet, Andy. Maybe reload?
Can you hear
Yes,
me, Daniel?
sounds good now. This is the fun, is it not, of online events? So while Andy is reloading, we will just be getting ready for the next part. Great.
Hear me?
Yes, I can hear you, Andy. So go ahead and share your screen and just go ahead.
Okay, cool. Thanks, Daniel. Okay, so let's share my entire screen. Okay, so can you see my screen now?
Yep, I can see the Jitsi window, and then that is perfect. Thank you very much.
Okay, okay, great. All right, thanks, Daniel. Thanks for the introduction. So yeah, I guess it's a real pleasure being here today. And I guess what I'm going to be talking about, I guess, is my art practice, I guess, and how it relates to complexity and basically how I'm using sort of complex systems. Almost the background behind it, almost the challenge, I guess, of using things like sort of complexity for a creative intent, almost like how I'm trying to address that problem, almost like how to actually work with these often quite unruly, really rich, interesting, emergent systems, but how to actually work with them creatively, almost like as a human, how do I actually work with this sort of material? So I hope it's going to be a sort of interesting talk, and I'm just looking forward to hopefully like discussion and questions that you may have. So to give you, I guess, a bit of a sort of background, I guess the real core of my work and I guess what really motivates me probably isn't complexity itself, but it's almost just ideas of this as being the sort of material that life uses, the sorts of things that nature uses, as being sort of complex systems with lots of like interconnected parts, of like, different chemicals that react with each other, or neurons that trigger each other. Or I guess the thing that I'm most of my artwork to date has been sort of focused on is, I guess, morphogenesis, so the idea of creating for sort of rich, what's basically a complex system, creating form. So what you have is these sort of these rich systems, but the other sort of critical function to that system, something that, almost like, how do we make sure that the system isn't just doing sort of crazy stuff, or isn't just out of control, but actually is being harnessed, which obviously in nature is sort of happening, which are creatively. It's like how do we work with the systems, which often actually these sort of complex systems, they actually don't do anything, or they just go completely out of control. And often it's just sort of like there's a transitional behavior where the really interesting stuff starts to happen. So how do we almost like find that sort of behavior, and then how do we find what's potentially the most interesting creatively, or things that I want to direct it towards, but not over direct it as an artist. So essentially, what is this medium like to work with, and how can I actually work with it creatively, I guess, is almost like the real sort of like core questions that I'm trying to work with in my practice. So to give you sort of an example, this is now showing sort of one of the works that I did a few years ago, about 7 years ago, called Cellular Forms. And what this is basically showing is a number of different animations, simplified model of cellular growth, actually just running on a GPU. So, and basically every simulation starts with a ball of 42 cells, and then there are rules for how cells divide. So things like light rays coming in from outside hit cells, and that allows the cells to generate food. And then there's rules for things like, does that cell keep all the food itself? Almost like, is it really selfish? Or does it share that food with its neighbors, almost be more cooperative? And then, it's what forces does each cell exert on the cells around it? And when a cell splits, which direction does it split in? Does it split in just a random direction, or a direction that's, where there's maybe most tension in the surface, or whether there's most curvature or least curvature, or maybe there's external forces and directions that can affect, the direction of splitting. So there's basically almost like a sort of mixture of all those types of ingredients in the system. And what I'm interested in is almost like having enough ingredients to hopefully create a rich range of different, behaviors and forms and structures, which is hopefully what you're seeing here, that basically this is all the same simulation system, but just by different strengths and of different influences, different parameters of the system, you often get like really surprisingly different sort of structures being created. Actually, all these simulations actually are saying they start at 42 cells. These are running to about 50 million cells by the end of the simulation, which is another thing I'm interested in, is almost like aesthetically getting to a level of detail and complexity, which I think is almost meaningful to us, where we start reading things in it, but instead of just being almost looking just like a ball of chemical cells, it starts reading as something of quite maybe ornate, organic complexity. And I guess I'm also working almost like then sort of sampling this by almost like working with like growing plants, or growing almost like growing seedlings. So I'm generally, first of all, generating a lot of examples of things to quite a low resolution, about 1 million cells. And then from those, I'm seeing ones that look like they might have some potential to be interesting. And, so almost basically I'm sort of rating and categorizing the sort of like things at the seedling level. And then I can do things like, a seedling which I think is particularly interesting, I could then create a number of mutants around it, or I could crossbreed it with a number of other cells, other sort of seedlings. So it's almost like sort of like a plant breeding sort of process of trying to sort of navigate that space of possibilities to hopefully start to find interesting sort of forms and structures, and also basically learn. So I guess that to give you a little bit more sort of detail. So what I've got in all those simulations, so basically it's a sheet of cells that's effectively, creating the structure. So basically there's these streets of triangles of cells connecting triangles, where like in this diagram, the black dots are the cells and the edges are basically links between the cells. So it's saying that each cell is directly linked to a small number of other cells, sort of like immediately around it. And then we have rules for cell division, so that basically after a cell has got enough food, the sort of like food level exceeds a certain threshold, then that cell is selected to be divided into two cells. And we do this by choosing a plane of cleavage, so that almost like choosing two edges, which is where the split is going to happen, and then deleting all the edges to one side of that edge, creating a daughter cell, I guess pushing the parent and the daughter cell a little bit away from each other, and then reconnecting the daughter cell back into the mesh. So basically what we end up with is, we start with a triangle mesh, a mesh of triangles. What we end up with is still a mesh of triangles, but now with an additional cell sort of added into the structure. And as hopefully you can sort of see, it's an incredibly simplified version of sort of biology. I mean, it's sort of like it's really plain, it's amazingly simplified. Instead of all the things in biology, like cells, then different types of cells being created and cells walking off into and finding where they should locate themselves and things like that. This is like saying it's an astonishingly simple model. And that's kind of one thing I'm often interested in is how simple could the model be, but yet hopefully still create surprisingly sort of rich results. So then the other thing I have in here is basically a set of influences as the other cells around it. So firstly, just like a set of almost like spring forces, so that each cell is tries to maintain a rest distance from the other cells that it's directly connected to. So if the cells are too close to each other, they sort of push themselves apart. If they're further apart than the rest distance, they'll try to pull back towards each other. Then the next thing I've got is another sort of two influences, which basically almost like try to make the surface either bulge or sort of smooth out. And I guess this was sort of informed, I come from a computer graphics sort of background. And this is sort of informed by, there's quite a lot of work back in the 90s on cloth simulations, and what they found was that if you just have straight springs in a mesh, what you end up with is a quite limited set of things. It basically almost only acts like stiff rubber come chainmail. If you want to get more rich sort of like folding, what you need to do is almost like things that actively make the surface fold or try to keep itself smooth. So this is trying to be informed by that, by having firstly an influence where if the cells are closer to each other than rest distance, so that basically sort of the sheet is in compression, then the cells deliberately try to bulge outwards. So basically what we do is we have normals to the surface, almost like vectors pointing straight out from the surface. And what we do is work out where would the cell have to move to in order to get back to rest distance if it was having to slide along the normal. And we work that out for all the cells around and just almost like a bit of an average. And that's almost like something that the cell tries to move towards that sort of bulging position. And then secondly, is almost like a thing going the opposite direction. That it basically just says if each cell tries to move back towards the average of the cells connected around it. So that's an influence that tends to smooth things back out. And if you still think about it, these are almost like working against each other. And that's the sort of thing I'm often interested in, is almost like ingredients in these sorts of systems, so that and there'd be different weights for almost like how smoothing sort of influence. So you can almost imagine that if I had a system set up with the bulge influence being really strong and the planar one being really weak, what you might have is a setup where it almost wants to wrinkle and fold like mad. Whereas if the planar smoothing influence was much stronger, maybe this system would want to keep itself much flatter, so that you end up with things maybe more like sort of bubbles and sort of balloon-like sort of structures. So it's almost like by hopefully just trying to get a good, interesting mix of ingredients that are going to have a number of different behaviors. And then the final influence I've got in here is what I call the repulsion influence. So this is almost like a really small, almost like electrostatic repulsion between the cells. So what I'm showing here, hopefully you can see the animation over the link, is basically two simulations, but with exactly the same parameters for all the influences, apart from on the one on the left, we've got this almost like electrostatic repulsion, whereas on the right, there isn't any. And so this is basically the idea is as the cells sort of fold in 3D space and come close to each other, so cells which are distant in the sheet, but in 3D space come close to each other, they just have a small sort of repulsion, a bit like static electricity pushing sort of the cells apart. And with that, what we end up with is potentially much more rich, interesting structures. Whereas without it, there's a tendency for this system just go out of control and just like cells pass straight through each other and it all sort of ruts influence that hopefully means it's more likely to get something that's sort of structurally coherent, a sort of meaningful sort of structure. So this is then sort of implemented just in C++ and CUDA, which is like a language to allow processing on the graphics card, which is like great for doing like millions of processes all in sort of parallel with each other. So what you're hearing is something like in real time, there's like a triangle mesh being created and different parts growing at different rates, and you're ending up with a sort of like a structure being formed. I guess I can generally create about 1 million cell structure in about a minute or two. So it's like, hopefully in a few hours I can have like a few hundred seedling level sort of samples from this simulation system. And the sorts of results I'm getting actually, hopefully are a genuinely interesting, sort of surprisingly rich, given how I think ridiculously simple this system is, sort of set of structures. So here, these are all about 50 million cells, and this, I guess, it's almost like it's almost like a sort of slightly brain-like, I guess, but but not quite, maybe more geometric, but it has sort of echoes of those sort of almost like brain-like folds. Maybe this one's a bit more like maybe a reptile skin, and this one's even more stretched, folded sort of skin. And this one's maybe more like sort of a coral-like form. And this one, I guess, maybe is reminiscent of fish scales, maybe particularly like shark scales under a microscope. And I guess one thing I'm interested in is not making anything that's specifically like anything specific form in nature, but things which maybe have echoes of multiple different things, and maybe so you have reminiscences. But I guess one thing I've been finding is almost everything that does make structural sense, even though it's such a ridiculously simple system removed from biology, seems to have echoes. It seems to be almost like it's either does nothing or it explodes, or it does something that makes sense, and it's almost remarkably familiar, maybe of 2 or 3 things from nature. Maybe this one's maybe a bit like a sort of lungs or internal organs. And this one I think is almost quite like, it's probably about the least obviously biological structural thing that I got from that simulation engine. But even that, I think, looks, there's people that say maybe it's a bit like almost like insect mandibles under a really powerful microscope. But also maybe it's a bit more geological, a bit like the sort of like rock formations you get underwater when sort of volcanic rock, solidifies really fast sort of underwater. So in that, it's maybe, thing has sort of biological, natural sort of like, echoes and reminiscences. So then I'm working with these, producing artifacts. So here's like an art exhibition I had with, but presenting them in almost like a number of different ways. So and that's one thing I'm interested in, is almost like the data from the simulation is almost the original sort of thing that's created. And there's no right way of seeing that. Almost like every way of turning it into an artifact, every way of visualizing it has different biases and different ways that different things you might perceive, and different ways that people actually seeing the work react. So things like this have got like a high resolution 2D prints and a 3D printed sculpture, and there's also sort of 2 projection screens with like quite big animated projections. Actually, one of the things of these is that there's actually an arrangement of mirrors between the screens. So the idea is that if you looked into the mirrors the correct way, it's actually realized the whole installation is basically a really large stereo viewer, so you could see the structures as 3D sculptures effectively. And what I found is that it's not just that 3D things are better than 2D. It's actually because the way that you engage with things, the way you act with things, you actually read things differently. Often I'm also showing things maybe like X-ray view of the same data next to almost like an external render of the external surfaces, and you see different things in each view. So that's just one thing I'm interested in, is almost like having a range of different ways of turning this into artifact, and maybe it's almost like playing with mixing those together. And I was working with almost like then making modifications, experimenting with the medium by almost like different, adding extra ingredients into the system or extra constraints. So this is a sequence here called plant-like forms, where what I was doing was, whereas there's generally spherical-like forms I was showing you previously, light rays to create food would come from all directions uniformly. In this, it was like, well, what happens if the light rays generally come from above? It's almost like a narrow set of directions. Well, just that exactly the same system, but just with that sort of just changed rule, suddenly you start getting much more sort of plant-like and sometimes almost like fungal-like sort of structures growing. So quite different sort of shapes by really just one very, very small change to the setup of the system. And then there's another system series called hybrid forms, which was then looking at almost like the simplest steps towards multicellularity, so that everything I showed you previously, all the cells in the structure had identical properties to each other. So there's almost like only one cell type in the whole form. And this, it was like, well, what happens if you have more than one cell type? Well, let's have the simplest version, almost like a ridiculously simple version of that, where we just choose two different cell types. You can say like cell type A and cell type B, and then and then other cells that can be almost like on that continuum between the two. So actually what you're showing here is 4 different visualizations of the data from one point, where the green blue sort of form is showing the different cell types. So the bright green parts are one cell type, the bright blue parts are another cell type, and the colors between the two, the sort of cyan teal sort of colors, are cells that are some way a blend between those cells. And just that very small change, instead of ending up with most things being these pollen-like sort of spheres, suddenly you're getting almost like things that look like rich sea creatures or embryo-like structures, things like that. So it was like in this surprising extra range of morphologies of structures, just from again, it's like ridiculously naive, simple version of multicellularity. And then the final sort of art series I'm going to show is one called the vase forms. So this was trying to experiment with trying to make forms which I could actually fabricate as sculptures. Where what we was wanting them to do was be sculptures that could be stand on something. So it must be vase-like sort of forms that potentially could have a flat bottom, so stand on things, and potentially also a flat top. So we potentially could use this as almost like maybe supports for tables or columns or something like that. So a sort of like different, almost like I was going to say, this was, I guess, an experiment in trying to steer the forms in a direction, but not overly controlling things. So what we decided to do in this was, firstly, because we were wanting these sort of like forms with flat bottoms, flat tops. Well, let's start by growing the cells as starting in a tube, where the cells at the top and the cells at the bottom are constrained to being in a plane, so they can move in that plane, slide in that plane, but they can't, but they still will stay inside in that plane. And then as the structure grows, the daughter cells of those cells at the top and bottom will also be in those planes. So what you end up is almost like a growing tube of cells that sort of stretch between these two planes. And then a number of other influences to sort of like, which were basically changing the rates of growth in different places. And the first of these was trying to make things which hopefully would steer things towards possibly being naturally 3D printable. So I guess one of the big sort of limitations in like simple 3D printing, what's called like fused filament deposition, is that if you've got too much of an overhang, you can't print it unless you build lots of additional support structure, which you then have to cut out and throw away. So well, the idea was, well, if the overhang angle almost in the local area of cells could influence the growth rate. So maybe we slow down the growth if we get beyond certain overhang angles. Maybe that's a way of almost creating a bias in the system. So not creating a strict engineering simulation, sort of like, it doesn't guarantee that everything will be 3D printable, but hopefully it's a bias that's affected by overhang angles, so that hopefully in almost the space of possibilities that this system is going to create, there are hopefully going to be quite a few forms which are naturally 3D printable without having to build extra support structures. And then once we've got the idea of then changing growth rates in part structure, maybe we can use that in some other ways as well. So I introduced it, but we wanted vaguely vase-like forms, so narrower at the bottom and sort of wider at the top. Well, let's just put that in as a vertical, something like rate change, so that cells towards the top of the structure can, generally split faster than cells towards the bottom. And also, if we don't want this structure to grow too far outwards, we can also use the distance of each cell from a central axis to change the growth rate. So basically, the idea of these, all the sort of, it's a bit like dressing different amounts of nutrients, I guess, in this sort of like space. Almost like I want the plant to grow more here and less there. So hopefully it sort of influences the growth, but without overly specifically directorially controlling everything. It's almost like I don't want to throw the baby out with the bathwater. I want to, I want to let rich emergent behavior still occur in the system, but in a way that I've slightly directed it to create these hopefully vase-like structures. So obviously this animation, so this is what I then got from some of those initial tests. And I guess I was looking for some rich surprises, and this really did surprise me. Basically, it didn't work. Well, it didn't produce forms which were going to be 3D printable without support structures in the way I'd hoped. It started growing these almost like, I think, really interesting, but often really filigree, branch-like, tendril-like, horizontal, like the sort of structures coming out of the forms. So I guess it's almost like a cautionary tale, or almost like, don't try to outguess complex systems. Or they often produce really incredibly rich behavior, but if you're trying to direct them, they will often go in a completely unexpected direction. But I think the programming, creating these sorts of branch-like structures, I think would actually be really quite difficult. So I think what's interesting is this is genuinely emergent. I really was not intending to make these types of forms, structures from this code, but almost the system was rich enough that it just happened, emergently. And the reason that I sort of looked into it is like, okay, what's happening? Was basically, there was first of all a bug in my code that I guess I'm creating food in cells based on this sort of simulated photosynthesis, so light rays coming in, hitting cells. But one thing I was doing was I was almost, because I've got these cells between two sheets, and then I'm growing more and more and more cells. I was deliberately shrinking the cells, shrinking the rest length as the sheet grew. But I was forgetting to do that sort of change of shrinking the size of the cells as far as the photosynthesis code. I hadn't sort of put that into that bit of the code. So as far as the photosynthesis, the light rays were concerned, all these cells were huge. So that these big cells, if one cell sticks out a little bit further than its neighbors, it tends to shadow all its neighbors and grabs all the food. So anything that sticks out a little bit becomes the focus where all the food happens. And then the second thing was how I'd implemented these varying growth rates for things like the overhang angle. I hadn't thought through properly. What I tried doing was trying to be a bit too clever. Always dangerous trying to be a bit too clever. I sort of thought, okay, simple way to do that is I could just change the rate of time. Almost like I can slow down growth by making time go slower in some parts of the structure than another. Oh, that's a fun, cute idea. Bad idea. Basically, what that was doing was effectively freezing the structure when it got beyond certain overhang angles. So essentially what I've done is come up with a recipe to cause growth where anything that sticks out a little bit further from the surface than other things nearby, And if anything goes beyond an overhang angle, it becomes solidified, it becomes a permanent structure. So essentially, it's a recipe to create, accidental recipe to create these like branch-like forms. So I said, useless to make actually fabricated sculptures, but I think really interesting in its own right. So it almost becomes an accidental sort of artwork series. So I've been presenting these in sort of stereoscopic installation. So here's an example. This is actually a wall-mounted sort of stereo piece. So it's basically sort of like 2 high-resolution screens and then 2 mirrors. And hopefully it's an interesting piece. If you're like in a gallery, it looks a bit almost like a slightly crazy computer triptych. But if you put your eyes at the place where almost like the viewing goggles are in the middle, basically, and then look through those, it's basically a mirror stereo viewer. So that you can then see this as an animating, rotating form with the forms growing. But it's a sculpture, 3D sculpture, but it's a virtual 3D sculpture. And then I went and looked at the code and said, okay, worked out why it was creating these crazy branches. It's almost like fix the code. So then, and then that's now creating much more, I guess, was what I more expected. So these sort of forms, which are these more vase-like structures, generally small, hopefully more 3D printable, but still hopefully having like a really interesting range of different structures and forms. And hopefully, it's surprisingly natural looking, but having these like variations of things like some areas with really incredible detail and some areas really stretched and smooth, some areas with these deep crevices and grooves, which also structurally may be really strong. But and then some areas almost like canopy-like structures, almost I guess when you hit certain growth rates where you start getting like real proliferation of cells, then you start getting these almost canopies. Again, something I hadn't expected, hadn't deliberately, I guess, programmed into it, but it's a behavior that I found from just experimenting with it, and I really like. 3D sculptures. So this is actually an exhibition from actually the Victoria and Albert Museum in London invited me to have an exhibit as part of the London Design Festival. So basically it's like an exhibit with showing some sculptures, and also I guess what was almost like developmental series, so small maquettes of the sculptures, showing them going from their original tube-like structure to the final sort of vase, basically going from about 300 cells to start with to about sort of 10 or 20 million cells by the end, and also animations showing the structures sort of growing. So I hope you can see I am actually be able to make these forms actually as sort of real sculptures. So now I'm going to sort of like switch into maybe almost like talking a bit more about the process and the backstory behind this, because I believe these systems. So basically it's the problem of like, okay, now when I've got a system like this, how do I find interesting behavior? How do I find things which I think is particularly rich, and things that I look at and think there's something of aesthetic quality, or something that a reason that I react to that form but don't react to that form. And I guess one intuition I've got is that it's often almost transition points in the behavior, the space of behaviors that you're always looking for, and looking for not so much typical behavior, but the sort of interesting atypical behavior that often happens at transition points. So this is actually a plot from work from quite a few years ago called the aggregation series, which basically is a plot of changing parameter values. So in this series, I just had a small number, 2 or 3 parameter values, almost changing the environment that things were growing in, basically using a very, or even simpler form of growth, what's called diffusion limited aggregation. So, but this is basically where along the horizontal axis, I'm changing one parameter of the environment, along the vertical axis, changing a different parameter. And I guess when you get in this, as you get to the far right-hand side, the system's becoming unstable. It's getting to the point where there are forces in the environment that are so strong that it's almost shredding the structures. And in this, it's almost the tipping point as you get into that instability, almost in the sort of 6th and 7th columns, where you start to see, I think, some interesting, rich, unexpected sort of structures, which potentially okay, that's something I think has some artistic qualities to it. So it's almost like, how do you then sort of find transition behaviors? Maybe those sort of transitions to instability. It might be, I guess in chaotic systems, almost like transition point between regularity and chaos, almost the border as you get into chaos, like the edge of the Mandelbrot set where potentially interesting behavior happens, or just almost like any other different behavior, maybe two different places in the space with different behavior, and maybe there's a boundary between the two where we get some sort of like hybrid interesting transitions happening. So I guess the problem was how to explore that. If you've got sort of 2 or 3 parameters, you can just do plots like I was showing you, the previously. But what happens if you get more parameters? So, and often these sort of cellular growth systems are getting these like 12, 20, 30, 40 parameters. So one thing I've been writing over quite a lot, actually, it's over the last 10 years, is writing a sort of piece of software called Species Explorer, which is basically just an interface for me to use to try to explore the sort of parameter space and the possibilities. So basically what I can use is this software it sets off, chooses parameters to try new samples to generate forms at, sets off the generation engine, and then it gets back the images from the generation engine once it's generated a form, and then I can look at that in the user interface, and I can do things like I can give them scores. So I can say, well, that one just failed. The simulation didn't run. I'll give that a 0. That it ran, but it was really rubbish. Let's give it a 1 out of 10. That's a bit more interesting, maybe a 3 or 4 out of 10. Oh, that's really quite interesting. Make it that 6 or 7. So basically just giving it subjectively a score of what I think almost like aesthetic value. But I can also give scores for other things like how many tendrils something's got. Or I can put things into categories. So I can say that those things all remind me of, furry structures. Those are all brain-like things. Those are broccoli. Those are sea creatures. I can give things almost like names and categories, which is hopefully then useful data to almost like help navigate the parameter space. And then I can use almost like a number of different methods to then set off generating another next population, that set of individuals. So I can do maybe just generate individuals with random parameters, which particularly when you're starting with a new system, I've got no idea what parameter values. I've hopefully put together a set of what I think interesting ingredients, but I've no idea what parameter values may work. Let's just try things randomly. And once I've seen a few things, maybe I could start using like evolutionary methods. So particularly if I've maybe found a form I really quite like, just maybe creating some mutants around it. So sort of like close in the space around that individual, randomly mutate the parameter values and generate another one, or crossbreed that individual with another one. So almost like taking a leaf out of evolutionary sort of methods. The other thing I've also been using is very quite simple machine learning. So it's basically a system what's called k-nearest neighbors, which is like a ridiculously simple form of machine learning where you just estimate values or categories based on what is the value of the nearest neighbors in the parameter space. So it's almost like I will estimate the score at a new point in the parameter space as being what are the scores of other things that I've already sampled nearby. So ridiculously simple form of machine learning, but hopefully gives me some level of prediction of new points in the parameter space, as long as I haven't got too many parameters. And so I've been using this as like a number of different techniques in here. Now, more recently, I guess one of the kind of stories in artificial intelligence is deep neural networks have become the thing. They're almost like incredibly interesting results from deep neural networks and training these on huge data sets. So I guess one question is then, well, can we start using deep neural networks to almost like take these ideas sort of even further? So this is something that I've been, actively working on the last couple of years, actually is a project together with John McCormack and Professor John McCormack and his Sensi Lab group at Monash University down in Melbourne. And I guess if you wanted to look at much more about this, I guess we had a paper in Evo Musart earlier this year, and also just a couple of months ago, an article in the Journal of Neural Computing Applications, which actually particularly that journal article has got some of our latest results. So if you're interested in finding out more, go and have a look at those. But just going to sort of talk a little bit about some of what we've been finding with this almost like new work trying to say, can we use deep neural networks? So I guess the motivation is, okay, can deep neural networks, well, previously, what I've always been doing is rating and categorizing everything myself, just by looking at the renders that come from the simulation engine. Can a deep neural net start to do that, or learn my preferences, learn what, how I react to things, and start to reliably, rate and categorize things if it can see based on the images, what you call the phenotypes. And also, I'm doing like these very naive estimates of, what the scores and ratings would be at new points in the parameter space using k-nearest neighbors, this very simple form of machine learning. Well, are there other things where could deep neural nets do better? Particularly if I can predict what the values are at new points in the parameter space, maybe that can give me us tools to directly navigate to interesting new places in the genotype space. So basically, that's sort of like the motivation. So what we've been working with is actually data from the original cellular forms, where basically we've got about just over 1,700 cellular forms, which I basically, from the original work, have rated and categorized all the forms into categories like animal and balloon and brain and plant. And these all have like 12 parameters in their sort of genotype. So that's basically the data set. And actually, we published this data set, so it's actually on GitHub, open source. People can, look at the data set and use it in their own research work if you want to. If you just go to the journal paper, we've got the links, to where the data set is. And so what we've been doing this. Well, firstly, one thing is, well, can it automatically categorize things? So using techniques, things like were called variational autoencoders and TSNE. Can a deep neural net almost automatically, without training from me, start to categorize things in a way which seems to roughly match maybe the sorts of categories and divisions that I'm making? And what we're finding is in genotype space, it really doesn't. That genotype space, at least all the techniques we're trying so far, are effectively dimensional reduction methods based on deep neural net data. Or deep neural net produces a vector which is then dimensionally reduced. It really seems to almost always just be a bit of a mess. But on the phenotype space, when it's actually looking at the images, we do seem to be getting things that seem to make structural sense. And almost this potentially could be tools to help, almost suggestions for how to divide the forms into different categories. So maybe almost like finer subdivisions, maybe than I've made, almost like interesting subcategories to make. So basically, the lower plot there, you're hopefully seeing that the categories that I put things into seem to almost like match reasonably nicely with what this, which is using TSNE to do dimensional reduction, seems to produce like quite sort of interestingly structural, meaningful results. And then looking at like, can it sort of like score things and rate things? Sort of similar to me, based on both the image data and the phenotype and the parameter values, the sort of genotype. So what we're using here is actually two different types of neural nets. So firstly, from the image data, we're using sort of ResNet-50, which is a reasonably state-of-the-art convolutional neural network used to classify images or classify and rank images. So basically, this is a neural net that's been or pre-trained on over 1 million images from the ImageNet database. So I guess it's almost been already trained how to see. And then what we can do is just train the last few layers of the network to say, okay, what we want you to do is rating and categorizing our sort of cellular forms data. And the second thing is using what's called a deep fully connected deep neural nets. It's where you've got like a certain number of hidden layers. So basically, we can set our own inputs, which would be the parameter values and the genotype, outputs being the scores and categories we're interested in, and then a small number of layers of neurons between the two, as a way of hopefully getting predictions based on the genotype space. And what we're finding is it does work. So basically, compared with k-nearest neighbors, where we're getting basically at best about a 50% prediction accuracy, there are things like ResNet-50, looking at the image data, is getting about an 87% prediction accuracy, and a fully connected deep neural network on the genotype parameters is getting just under about 70%. So deep neural nets seem to be already doing better significantly than we were getting with k-nearest neighbors. So I hope that's useful. And then what we're looking at is integrating this into Species Explorer in a sort of number of different ways, almost to hopefully be tools to help explore the space. So the first one is almost like what we call a categorizer. So the idea of this is it's showing us what categories it would be putting things into. but also showing us the confidence levels, so that basically with the green levels on the left-hand side are where it's got something saying it's got high confidence that this is the category it's putting it in. It's got the category correct. Whereas the things on the right-hand side and the blues and purples are things where it's saying this is the category it thinks it's in, but it's not that confident. It could be close to being in a different category. So what the idea is that hopefully with a really quite small amount of training data, we'll be able to see how reliably it's working and what we need to maybe how to correct it, which things it's getting wrong, and also maybe to confirm things it's got right, but really quite quickly correct where it makes mistakes, then retrain it again, hopefully with a relatively small amount of training data, get it to the point where it's like really quite reliably sort of categorizing things. And then we can use this almost like to automatically look for transition points. So the idea is here we can check like two forms, maybe like here, like form on the left is what we categorize as balloon-like, form on the right is what we categorized as brain-like, and then it can generate almost just a simple binary chop search. They can like generate the form in parameter space halfway between the two, and then generate that form. The ResNet 50 can look at it. It decides, oh, that looks like another brain-like form. Okay, now let's look halfway between, now the new sort of almost half range, and it just can keep on narrowing down, hopefully getting closer and closer transition points. I guess there might not be a transition point between these two categories, but if there is, hopefully this is a sensible, simple approach to try to find a transition point automatically. So I can set this off overnight, come back in the morning, and hopefully I'm seeing examples like this where it has hopefully automatically found a potential sort of hybrid transition, which may well be interesting. And the next thing is almost like directly showing us almost what the landscape looks like, so in the genotype space. So this is what was like showing 2D sections through the parameter space, but with predictions from the neural net, where it's predicting, so almost like the red areas where it's predicting, this we a balloon-like form. The light blue is what's predicting a brain-like, and yellow is where it's predicting animal-like. So I guess almost this area between the red and the blue is hopefully a transition zone. It might not be, because it might not have enough data to be well trained yet, but I can just in this interface then click on that position, generate a form at that, have a look at it. So hopefully I'm also getting to see and understand the landscape. So almost like mapping tools to help explore the space of possibilities. And finally, this is actually some recent work. So where we're trying to generate some forms which actually potentially might be fabricated at room scale, but with almost like tree-like and root-like behavior. I hope you're seeing we're actually hopefully getting like a really quite interesting mix of, almost like hybrid-like sort of behaviors sort of going on in some of these forms and structures. Okay, so I think that's the end of my presentation. Hopefully we got time for some discussion. So I just stop sharing my screen.
Cool. Awesome. Thank you so much, Andy. That was really an exceptional talk, and there's a lot of cool stuff and also some great comments from the participants. So first is just a note, and a question is, some of the videos were a bit just choppy, which is all good. Other than your site, the videos tab, is there anywhere else where people could go look at the videos?
Yes. So there's, I think most of those videos are on my Vimeo channel. So if you just go to Vimeo, Andy Lomas, or one word, that's my Vimeo channel with almost all those videos. Yeah, I think, trying to think which video. Oh, I guess the only video probably wasn't shown there is the one where I'm just showing the with and without the forces between the cells, but all the videos of, and actually things like the hybrid forms video, I think is actually really interesting, which I probably would have showed if I had more time. So yeah, go to my Vimeo channel, to Vimeo/Andy Lomas.
Great. We'll include that in the video description as well. So here are a few questions from the audience. So one question was, what is happening inside of the vase-like forms when you're looking at them or other forms like with an X-ray from the inside? What is that like?
Again, actually, I guess definitely look at the Vimeo channel. I could try sharing a video right now if you want. The very different, I think, is the answer. Is you see very different structures, myself see internal things, and what's surprising is what you could see on the outside often disappears. So it's actually, it's not, you perceive very different things. Actually, if you go to my Vimeo channel, there is examples of those. And basically, you have a look, it's like have those cellular forms, and they have cellular forms X-ray, which is like the X-ray version of the same thing. And actually, this is also something I've been doing some work on recently. I've been actually with some of the latest GPU hardware. There's hardware acceleration for ray tracing on some of the latest GPUs. I'm actually now able to get rendering in real time, which makes you can have like a stereo viewer which you can actually interact with, which I think also gives you another level of depth perception, including both X-ray and these sort of external views. And from looking at like an X-ray view in a stereoscopic view that I can actually manipulate directly, I've just recently seen structures in there which I wasn't aware of myself. So yeah, it's almost been revealing, yet new things which was already there in the data, but just because I hadn't been able to perceive them with enough dimensionality, I wasn't aware of previously.
Perfect. And also, you don't see it on your video side, but I am showing on this recording videos from your Vimeo on the screen. So people are getting to see them. Cool. So the next question was,
Okay, cool. Yes, I hope you can see on the X-ray recordings there.
Yes. How do we keep track of the feedback?
Sorry, I lost.
How do we keep track of the feedback going on during the embryonic growth simulation that you showed? Can we track positive and negative feedback loops as they interact?
So I just lost the end of that question there.
So how do we model feedback loops during embryonic development?
So can you say that again, Daniel?
How do we model the feedback loops in the embryonic development?
So how do we model the feedback loops? I'm trying to think of what I was saying. So one thing is I'm generally, actually all these simulations, I'm not intervening once the simulation actually starts running. So I'm sort of just choosing parameter values in the parameter space to, using the species explorer, things like that. So almost like new parameter genotypes to try out. But once I'm setting off a simulation, I'm just almost like letting it run and then choosing ones that I like. So it's almost like just letting a seedling grow. I mean, I guess there's feedback loops just inherently in the simulation engine. Things like, because the cells are all affecting each other, that, where each cell moving to different positions then causes, cell A affects cell B and cell B is felt cell A, and, there's lots of interconnectedness, which I guess is almost like the essence of, having sort of, interconnected non-linearity and having iteration of multiple time steps as being almost the essence of most complex systems. Does that sort of answer the question?
Cool, great answer. Here's another question. Some of these projects were visually beautiful, but also required significant computational power, like you mentioned, graphical cards and other things. And you also had the transition point plot that was like a search in parameter phase space, but it was being evaluated artistically. So just from these projects, what insights would you have for how interdisciplinary teams could be working together, or how there could be a fruitful discussion with machine learning and aesthetics and complexity? How can we facilitate that conversation?
Yeah, that's an interesting one. I mean, I guess, I do what I'm doing because I'm interested in it, if I'm really honest. I mean, I guess the artistic, people seem to be interested in, exhibitions and me doing things as artistic artifacts. And I guess that gives me a certain freedom to play. And I guess the direction of, I don't have to justify myself. It's just, what I find aesthetic and interesting, can drive it. But there was one conversation I have had is, with people much more from a pure science background. Recently I was doing some work with together with as a life science institute up in Dundee University. And actually, they've actually got an art exhibition space in the Life Sciences Institute. So basically, I was involved in an exhibition there. One thing we did was there was a researcher, as a physicist seconded to the institute, who's actually doing very similar almost process simulations of cell growth. But one thing that was interesting was like the similarities and differences between what we were doing. So Rusko was guessed, that this simulation simulations were totally true to the biology, and it was obviously a key and important. So often doing things to much lower levels of cell counts, but because you're trying to do things as accurate as possible. Whereas I'm generally just trying to do things, doing millions of cells. I mean, one rule of thumb I've always got is that you've got to do at least 1 million of things before it becomes in some way psychologically, visually interesting. So I was always, avoiding accuracy in favor of, just being able to do computational amount of data, amount of complexity. And also, I guess the thing where usually, I guess, on the science side, you're more almost trying to categorize sort of typical behaviors, whereas I'm often more just diving into the canyons, almost the atypical behaviors. Not that you always have to go that direction in science, but maybe that's the sort of just general tendency, is that is that you're drawn to wanting to categorize the whole space, where I'm going, whoa, here's a deep rabbit hole that I think is particularly interesting. So yeah, I guess it almost gives you permissions and different directions to go, but maybe there's some interesting dialogues that can come from that. And actually, one thing we did was we actually almost created our own work. We did a thing where Rusko commented on my artwork, and I also then curated some of the Rusko's science simulations, which we edited together as a video to go in the art exhibition. So it's almost like, artists commenting on scientists and scientists commenting on artists.
Wow, thanks for that. That's really a beautiful metaphor with the parameter space and what the artist sees and what the utilitarian sees. So here's just one final question before we adjourn to the Jitsi link that the RSVPs have. This last question is about just change in time, kind of a feature of complex systems. How has your aesthetic perception changed over time while you work with these forms? So when you rate them or just in playing with them, how do the way that they interface with you, how does that change through time?
Sorry, could you actually say that again? So the sound's getting a bit choppy.
Oh yeah. So just how do you develop in your relationship to making and viewing these forms? Like as you work with one shape family or tool more, how does your aesthetic perception change?
Oh, okay, yeah. I guess that's a good question. That it's almost like you're going into a new system, and it's almost terra incognita, or maybe almost like a fear that you've just misprogrammed it, and is it going to produce anything at all interesting? And I guess then that's one of the things, I guess, is that it feels almost to me like you go through a number of phases. That there's almost like sort of like a initial, almost like random exploration. It's almost like you're parachuted deep into the forest, and you've got no idea what's around. And then after you've found a few and you start to find this is swampy area you don't want to go back to, it really doesn't work around here. So it starts almost like maybe just getting very broad ideas of what possibly works at all, but also lots of things that just don't work and you should avoid. And then you start maybe saying hints of things which are maybe more interesting. And then you maybe want to focus on those and start to find those, and find what, is this almost like a niche that you want to explore more. But then once you've seen, you've found like, hey, it looks like there are cauliflowers over there. Oh, that's interesting. After you've generated a few hundred cauliflowers, you're getting really bored of them. So it's then you're actually interested in novelty and things like that, or what. So you actually almost are going through different phases, which is one of the I almost critique, I guess, of classic, treating this as an optimization, treating this, or the classic interactive genetic algorithm tools, things like that. To my, it's particularly as you want your intention changes, and it might just one day to the next that is I'm actually interested in, like, I'm kind of close to an exhibition. I want to refine things to get a neat. That looks pretty interesting. It's pretty good enough, but are there even more interesting things close to it? Or I want to do a family of those all together. Which might want to almost like very different tools from. Okay. Is there anything just completely, utterly new out there? I want to see, and genetic techniques often tend to get trapped in niches, to my mind. So that's okay. So your relationship with the system really does change over time, and maybe later on you start maybe becoming a bit too precious of it. It's like you don't want to change things because you might break it and lose what you found that's already cool. So you then want to start with a new system. So it's something you learn. So, and it's learning. It's like working with clay or working with paint, a new type of paint or something like that. That you learn how to work with the medium. So yeah, it very much is a progress, and that's one thing I'm interested in, is how as me as a human working in combination with the computer, I need the computer to do these simulations, but I also need the computer almost as my collaborator to help me explore, work with the medium. Particularly, I guess in my mind, it's almost like making me brave enough to work with even more unruly systems. It's almost like I'm worried if I don't have the computer helping me, I will try to be too tame, and I want to be a bit more fearless. And hopefully these things like using, machine learning and things like that will hopefully allow me to be even more fearless, which with working with these sorts of systems. And the more unruly it is, hopefully the more rich, maybe the more difficult to find interesting results, but hopefully the richer those results might be.
Thanks for explaining that frontier to us. Just one last quick question. Somebody asked, what is the name of the scientist at Dundee University? Is there a collaborator at Dundee?
So you're asking who the scientist was?
That's what somebody asked, yeah.
So just trying to find Rastko's surname. Dr. Rastko Sknepnek. Have I got? I don't think I've got chat on this, do I? Or do I have chat?
You can send a chat.
I can spell it for you.
You can
it's
spell
Rastko, Rastko.
Okay, we can definitely do the chat. But thanks for the question. And while you're typing it, thanks again, Andy.
It's Sknepnek. Basically, Dr. Rastko Sknepnek at the School of Life Sciences in Dundee.
Perfect. Well, Andy, thank you so much for this fantastic talk. Gave a lot of us a lot to think about, and we will adjourn now to the Jitsi that we have provided everyone with. And thanks again for watching it live or on replay, and we will see you all in the Jitsi in a couple of minutes. Thanks again, Andy. Bye.